Hello, everybody. I am Dr. Shaima Kassab, Associate Professor of Medicinal Chemistry. Today, I'm going to teach you how to efficiently enhance the potency of KRAS small molecule inhibitor. As you see on the screen, this is a complex crystal structure of a small molecule KRAS, a small molecule inhibitor, co-crystallized with the active binding site of KRAS oncogenic protein. When you talk about when you plan to efficiently enhance the potency of the compound, the first thought should come to the mind is to force the compound to form more bonding forces or interaction forces with the amino acids comprising the active binding site. As you see in the complex crystal structure, benzene ring in the beginning of the structure here forms hydrophobic interaction with leucine 56 and very nice and strong hydrogen bonding formed with in the middle of the compound. In the middle of the compound, there is nice and strong hydrogen bonding force formed with glutamic 37 via NH of the carboxamide linker of the small molecule inhibitor. The rest of the compound involving terminal amino ether side chain failed to interact with any of the amino acids which are close to this region of the molecule, like serine 39, glutamic 37, and methionine 67, glutamine 70. So you have the best decision. To my knowledge, the best, and from my experience, according to my experience, the first thing you have to think of to enhance the potency is to modify the structure in this region, to introduce different pharmacophore, a pharmacophore to be able to form any bonding force or interaction force with methionine, either methionine 67, glutamine 70, glutamic 37, or serine 39. So let's work on removing this. Also, that for the another thing you have to be aware of is to replace this amino ether side chain with heterocyclic fragment or aryl fragment containing hydrogen bond donor or hydrogen bond acceptors, but give up from the chemical library, give up of introducing similar, or even if it's not similar, aliphatic side chain or open side chain, because as you see, and according to the conformer of the compound of the least energetic state, as you see, the conformer of the compound didn't help this aliphatic side chain to stay in a way or another to form any hydrogen bonding with the amino acids of methionine 67, glutamine 70, or serine 39. So you have to replace this amino ether side chain with any other pharmacophore with hydrogen bond donor or hydrogen bond acceptor, heterocycles, either alicyclic ring or aromatic rings. Building up your database library or chemical library you are going to use for introducing different pharmacophores other than amino ether side chain should be carefully designed to achieve the goal of the mission accurately and precisely. At the same time, diversity is very, very, very important to give you possible opportunity to choose to prefer one compound and or to involve one compound, or to filter out one compound and exclude the other. It depending on your eyeballing, your ADME profile, the ADME profile you would like to choose. Also, compound like ligand properties. It's very important here to look into recent resynthesis scores. It's 61.54. Topological polar surface area, log P, it's not the ideal value. It should be between for drugs, it should be between 1.5 to 3. For the solubility, it's nice. So you have to introduce the different pharmacophore, observing the changes that would happen to the ADME profile, physicochemical properties, topological pore surface area, if you get a compound to achieve the goal of the mission, and at the same time it didn't alter the numerical value of the original ligand, or improving the numerical value that was not optimum in the original ligand. All these considerations would be nice to be aware of. Let's start with removing the amino ether side chain, which are going to be replaced by different pharmacophores in the form of heterocycles. Let's check also that this region on which we will introduce the diverse pharmacophores to interact with these amino acids. Let's check that this position is among the R vectors. For the arrow headed up here, it means that this position is available to get substituted by different substituents. So you have to check this carefully before starting the process. Let's add group to ligand. You have to choose a carefully designed library instead using random and general library to achieve the goal of the mission of development of the compound. And at the same time, you make sure that the compounds which will be filtered off are diverse. We'll remove the original library and add our library. Let's see, we'll minimize the energy using the scoring function, this one which is reported, and start searching for different pharmacophore that could be introduced in this position, and check later which one of the chosen pharmacophore will be able to form hydrogen bonding with glutamic, serine, or glutamine 70 or methionine 67. Now 10, 11. Here is, as you see, here is the linker or fragment in the chemical library, which is introduced to the position we have chosen for replacing the amino ether side chain with this heterocycle. These heterocycles are commercially available, and it has plenty of hydrogen bond donors and hydrogen bond acceptor atoms to give us the results we are dreaming of. Here is the out molecule after inserting or after introducing the fragment to the position we have chosen in the original ligand. Here is the resynthesis score. Now we have a little bit lower, but not bad. 48%, not bad. A little bit lower, but not bad. It's still accessible to synthesize, to resynthesize. Since these fragments are commercially available, so you can insert this group to the original ligand in a very fast and easy way. If you already are working on this ligand and you synthesized it, eyeballing is eyeballing here is important because sometimes you don't prefer to use to involve a certain linker and you prefer other linker, something like you prefer to use to insert triazole and you do not prefer to use say thiophene group. Sometimes we find some fragments are quite more expensive than the other, and both of them achieve the goal of the mission. They form the same hydrogen bonding. So eyeballing should work here, and the decision is yours depending on how do you see the compound after inserting or after introducing the new pharmacophores to the original ligand. Let's check for saving time. Let's check what we got. We will change the color of the molecule after introducing or after inserting the new pharmacophore. Let's we can to we can sort the resynthesis score into descending order to make what comes first is the molecule with the resynthetic score higher than the second or the next one. Um, Now we can browse after reordering. The first one will be of the highest synthetic score or synthetic accessibility. Mhm. Let's change the color. This is the new compound. As you see, it has the same conformation with the whole ligand, and except for and this one is free. This fragment, the newly introduced fragment, is free to position itself according to the best conformer or the best energetic state of the conformer. Uh, let's check which one was successful, which ones succeeded to form the hydrogen bond we plan or we expect to see. Yeah, here we have nice compound, nice conform, nice modified structure or developed structure, which succeeded to form further bonding force as strong as hydrogen bonding as strong as the one which formed by the original ligand with the same amino acid. This compound is nice, and the resynthesis score 48, it's not bad, it's nice value and agreeable value or acceptable value, not not very far from the value of the original ligand, which was 61. And I think this one is very nice and it's worth it trying with it. Let's check also for the other filters. I look forward to seeing a developed compound which is able to form a hydrogen bond with serine 39. This one didn't add something new. Okay. I am looking forward to see a compound that is able to form hydrogen bonding because this will be a kind of diversity, structure diversity. Yeah, here we have something we have a compound is able to form hydrogen bonding with methionine 67, which is nice. Also nice. So it's a kind of diversity, new binding pattern. Um, I'm still looking for a compound that is able to form a hydrogen bonding with, oh, we got it. Very nice. Now we have a compound that is able to form bonding force, hydrogen bonding force, with agreeable strength with serine 39. Now we have a compound is able to form a hydrophobic interaction with leucine 56, hydrogen bond in the middle region of the compound, hydrogen bonding force, hydrogen bonding with the glutamic acid 37, and where in the rest of the molecule was able to form a hydrogen bonding force with 39. I think this compound is very nice too and worth it trying with it. Let's check with this the ligand property. We have to remove the original ligand to see. Now we have not very bad. I think this compound is nice because this developed compound is very nice because the aqueous solubility was almost optimum because it's not far away from the optimal value. The optimal value is -4, so I think it's acceptable, not very bad. Log B got improved, which is also nice. The resynthesis score, not very bad also. So this compound, it's worth it trying with it. And let me notify you or emphasize that resynthesis score is for the compound as a whole. But if you have this part of the compound, and you just add this fragment, it would be nice and in a very fast and also in a very fast and easy way you would develop your structure. Here, I think upon looking for more options, you will find more diverse compounds with I think with nicer bonding forces in this area. This area kept silent and without interaction forces with the original ligand. After developing the structure, you made it a very active region, and it forms bond interaction force, and in this region there is bonding force formed with the bonding force 1 or 2 with the new ligand or developed ligand. I think upon checking more or looking at more or browsing more filters, you will see nicer uh, bonding forces or binding patterns with the developed compounds. Yeah, here we have also nice bonding force with the same amino acid, glutamic acid. Yeah. The resynthesis score is getting lower because we sorted the browsing in a descending. Here we have also nice bonding force with methionine. It's a strong bonding force, hydrogen bonding force. Yeah, here we have 51 score, which is nice, but it didn't form the bonding force we planned to. It made internal bond, the hydrogen bonding force, but it failed to form bonding force with serine, glutamic, or methionine, or glutamine. For glutamine, I think it's a little bit if we succeeded to, if there is a fragment or there have been a fragment to come from this position to twist uh, under the axis of the whole molecule. I think this one would be able to interact with glutamine. You can check for the whole library. Can check for more compounds. Yeah, here we have another compound. This is nice because now we have two compounds. I think the first one was, I think, imidazole or triazole. I'm not remember. I do not remember. But for this one, it's oxazole ring, and it forms hydrogen bonding force with serine. I think this one is nice too. Now you have options to choose one and leave the other. Here we have two more bonding forces, which is very nice. This one is very nice. And for the pharmacokinetic or for the log B value and log S value, I think it's not bad. Yeah, here this is the best conformer because as you see, it's much stronger than the original structure. It's the hydrogen bond formed with glutamic 37 with the original structure or the original ligand. This one is very nice. This compound is very nice. In the same way above the axis of the molecule, if we got a conformer with a new fragment to bend in beneath the compound or under the axis of the original ligand, I think it will succeed to reach or become closer to the glutamine 70. Um, I think this one is with very nice score, a numerical value for the recent. This is a score of chemical, in other words, chemical accessibility. In the pharmaceutical industries or drug industries, if you plan to patent the developed compound, the chemical accessibility and the resynthesis score is very important, which is if a parameter you have to be aware of the synthesis score. We have also here a compound with bonding force with glutamine. For saving time, I will. I think now the whole idea is clear, because it's still looking and searching for more linkers or more fragments in the mission. It filtered out 367 structures, and the searching will continue. And I think it would take time to, because of the chemical library is large. But I think the idea now becomes very clear to you, and I hope you enjoyed this session and learn it a lot from it, how you think of if efficiently enhancing the potency of a small molecule inhibitor into its active, upon looking into carefully into the chemical structure of the small molecule inhibitor side by side with the amino acids in the active binding site, comprising the active binding site. Um, you have to be aware of chemical structures and the chemical properties of the amino acids, as well as the chemical structure features, which position, which fragment you have to remove and replace it with the new fragment. I hope you enjoyed this session, and see you in another session to learn a lot in the journey of drug discovery. Thank you so much. Goodbye.